Hello there. This is the last bit of our notes from chapter 17. Um, so in this part of in this video, I'm going to talk about mutations. You should have a good understanding. You already finished um, your impact of my mutations handout, but I just want to clarify a little bit of terminology and make sure you understand which um, events are often more significant than others. So I'm here in your notes. So mutations are, a common misconception is that mutations are bad, but they're important because they are the source of all new genes and then also the source of all new alleles. And they're important because any change in the DNA sequence is going to potentially change the protein sequence, amino acid sequence, which can change the protein structure, which can impact function. Now, there's two different categories. You can think of them as events, things that happen that result in changes in DNA. And again, a mutation is just defined as a change in DNA sequence. Now, there's two basic categories. The one category are substitutions. This is just when one base is substituted for another. So it doesn't end up changing anything downstream because it doesn't change the number of bases. But then you can also have something called an insertion or a deletion. Sometimes those are separated out into two different categories, but those are events, and they cause what are called a type of mutation called a frame shift. So now let's look down here at this diagram and let's see if we can understand the difference. So point mutations are mutations that occur at a single point, and here where we have a substitution. So for example, here this base changes and then it's going to change the amino acid. So it depends on the amino acid that gets switched out. In the case of sickle cell anemia, it's pretty significant, but often it's not significant because it doesn't cause any changes downstream. But frame shift, and again, it also depends on where that mutation occurs in the DNA sequence and then how that translates where it occurs in the amino acid sequence. So this is a three-dimensional drawing of a protein, and you'll see here that if there's a mutation out here in the body part of the protein that doesn't significantly change the way it's shaped, or maybe change the way it's shaped at all, that's not going to change the function of a protein. But if that mutation occurs at the active site or the binding site of whatever the activity of the protein is, that's going to cause a significant change in the protein functionality. So the location of the mutation is important. Now let's go down to frame shift mutations, and let's, by way of looking at language, let's look and see how a frame shift mutation is going to change the DNA sequence. When you have a substitution, you can change the meaning of it. So for example, if I substitute in a C for a B, if I'm thinking about a three-letter words, again, thinking of the triplet code, or not again, but thinking of the triplet code, sort of translating that into three-letter words in English, you might change a sentence that said the bat saw the dog or the cat saw the hog. Those would be changes, right? Substitutions, but again, you still have this language that's the same. So you might have a similar structure. This is what occurs with sickle cell anemia, like I said, so it can be a problem, but often it's not a problem. Now, when you have deletions or insertions, you get a whole frame shift mutation. And so everything downstream changes it. So for example, if the B is deleted, or the C is deleted here, if it's showing this second line of code, you end up with everything being quote unquote wrong or changed anyway. And then same thing with an insertion. This is showing you see how all of this downstream is changed. Nothing upstream changes, of course, but it's the downstream that causes a problem. Okay, so we've talked about different events. An event is a point mutation. A frame shift is an event. Both of these are events. And substitutions and insertions or deletions. Substitution is an event that is a type of point mutation. Insertions or deletions are events that are type that cause frameshift mutations. And then we can define the types of mutations. So types of mutations are things like missenses or nonsenses. A missense just means there's a silent mutation, meaning there's a change in DNA, but there's no change in amino acid sequence, and that's because of the redundancy in the code. Think about it. Some of those 3-letter codons can code for the same amino acid. If we look up here in the codon table, if you have, for example, in this particular triplet code, if you have a U that is substituted for a C at a point mutation, you still get phenylalanine. So it's a silent mutation, and that silent mutation doesn't make a difference. However, in a missense mutation, you get a change in the amino acid sequence. So a missense this is not the proper definition of a missense mutation. So a missense mutation is when a mutation is a type of mutation where you get a change in the amino acids. And so that's what we have with sickle cell anemia. That's significant. A nonsense mutation is where you get a change in the amino acid sequence, in the codon sequence, so that you get a nonsense, so you get a stop codon, and it'll end the protein early, and you often, in that particular case, you have a pretty significant change. An example of an insertion and deletion is in cystic fibrosis. We have a triplet codon deletion, and it causes one amino acid to be missing, and it causes significant difference in the shape of that chloride pump. And so the pump doesn't, the chloride channel. And so the channel doesn't properly move chloride ions. Now, if we were thinking, just connecting this idea of our last unit, thinking about what chromosomes, where chromosomes were on, where genes were on chromosomes, that CTFR gene is on a chromosome number 7. So close your eyes and think about, okay, I have 23 pairs of chromosomes, pair number 27. If you are, if you're looking for the location or the address of the CTFR gene, it's going to be notated like this. So 7q31.2, which means Q means it's on the long arm of the 7, and then it's located at 31.2, and the numbers, the whole numbers refer to the band, the G band, and then that's the staining band, and then the decimal refers to which subband within that darker stained area if you were to do a staining for a karyogram. Okay, that's it for mutations. We'll talk more about mutations in chapter 18. Thanks for listening. I hope this was helpful.